Today's topic is mechanism of apoptosis. Apoptosis is a programmed cell death, thereby where the enzymes inside the cells get activated and those enzymes destroy the cytoplasmic protein, the nuclear protein, and the DNA inside the nucleus. Since the enzymes are produced by the cells itself, that's why we call the process of apoptosis as suicidal programmed cell death. The process of apoptosis begins with the activation of the enzyme called as caspases, as in the animation character Casper. The enzyme is called as caspase because this enzyme cleaves the protein after the aspartic residues. The activation of caspase protein depends on the balance between the pro-apoptotic proteins like Bax-Back beam and the anti-apoptotic proteins like BCL2 and BCLX. The pro-apoptotic proteins are the apoptosis inducing proteins, whereas the anti-apoptotic proteins are the apoptosis preventing proteins. The mechanism of apoptosis begin with the initiation phase and goes to the execution phase where the destruction of the cell occur. Now if you look at the initiation phase can begin inside the cell, that is inside the mitochondria, or the initiation phase can also begin from outside of the cell, from the surface receptor, which is also called as the death receptors. Once the initiation phase begins, uh, the there is going to be the stimulation of the execution phase where there is going to be the destruction of the cell. The mitochondrial pathway or the intrinsic pathway of apoptosis begins with the triggering stimuli like the growth factor withdrawal or the absence of survival signal or protein misfolding or the DNA damage. Now, these apoptotic triggering stimuli are going to result in the decreased amount of the anti-apoptotic proteins like the BCL2 and BCLX, whereas it is going to result in increase in the pro-apoptotic proteins or the apoptosis inducing proteins like the Bax, Back, and Beam. So when there is increased amount of the pro-apoptotic proteins, that is going to result in the increased permeability of the mitochondria. That is, the mitochondria is going to get leaky and is going to result in efflux of the cytochrome C. Now, when the cytochrome C leaks out of the mitochondria, the cytochrome C goes and binds with the apoptosis activating factor, which is also called the APOP1. The cytochrome C and the activating factor, these together form a complex which is called as apoptosome. Now, the formation of the apoptosome is going to stimulate the formation of the important protein which is called as caspase 9. The caspase 9 and the inhibition of the caspase antagonist, together they are going to drive the mechanism of the apoptosis into the axon phase, which is also called as the execution pathway. The dying cells have receptors over the surface, which is called as the death receptors. Now, these death receptors have cytoplasmic domain, which is called as the death domain. Now, these death domain have the adapter proteins attached to it. When the death receptors or the surface receptors, that could be either the T1 TNF receptors or the FAST receptors, bind with the T lymphocytes bearing the FAST ligand, there is going to be the activation of the adapter proteins via the death domain. The death receptor pathway or the extrinsic pathway, apoptosis, uh, we see there is the activation of the death receptors. The death receptors are attached to the cytoplasmic death domain, and the death domain are attached to the adapter protein. When this adapter protein gets activated, a signal is created that activates the caspase 8 and caspase 10. The caspase 8 and caspase 10 protein drives the extrinsic pathway of apoptosis to the execution phase. Okay, the caspase 8 can also result in activation of the proapoptotic protein called as the BID. Now, if the BID gets activated, that is going to drive the cell, the apoptotic mechanism into the mitochondrial pathway. Thus, what we see is the extrinsic pathway can also activate the intrinsic pathway as well. The final the phase of action, which is also called as the execution phase, begins with the activation of the two caspases named as caspase 3 and caspase 6. Now, once there is activation of these two caspases, caspase 3 and 6, that is going to result in the stimulation of some important proteins called as the endonucleases, DNase, and protease. These enzymes now break down the nuclear chromatin, DNA, and cytoskeleton respectively. The dying cells secrete some factors, and they also have a phospholipid flip. So what is phospholipid flip? Phospholipid flip is normally the phospholipid is inside the cell membrane, but the dying cells have phospholipid outside the cell membrane. Now these phospholipid and the factors secreted by the dying cells act as the attractant for the macrophages. Now these macrophages, as we all know, are the phagocytic cells, and so these phagocytic cells comes and clears up the dead cells. Thank you.